I'm going to do a test, okay?
Okay, that's fine.
Okay. Okay.
Hey, Stanislav.
Hey.
Do I sharing, right?
Yeah, you're sharing.
Okay.
I'm using my headphones.
So maybe. Okay. No, maybe not. I don't have.
You could open it up on your phone, maybe, if you want to read your notes.
Yeah.
Present on.
Okay.
Okay.
It's because I don't want to forget something, but really we can we can speak about that after.
Sure.
Fine.
But your camera is over here. Camera's up there. And then I switch my speakers to.
We good?
I think we're good. Maybe.
All right. Hey, Daniela, can
you hear us
Yes,
right now?
but not so good.
All right. Can you hear us now?
Yes.
I just muted myself.
Okay.
Wait, I see our talking icon on the screen. Like we're not muted.
Assume you're not muted.
Okay.
Do you use the Apple one maybe? Are you trying to do volume?
No, I'm just trying to switch the input.
Hola, Marco.
My 2 minutes or 3. Because I want to use those speakers.
I see.
So, do the thing.
Oh, I think you just need to turn off mute on the TV. I don't
Is
think it's
it
an input issue. I
Well,
think we just muted the TV because of all the meetings.
we muted TV,
So
so just turn it on. You know what?
while we're also 2 months this
So
is probably
let's
the
get
biggest story in
started.
our
So what's the problem running into right now?
I think we should just use your.
Just use your
Yeah,
computer audio.
All right, it's just good. Right there. Okay, we're ready. Yes.
Can you hear us, Daniela?
Yes, I can hear you.
I think everyone can hear us. We can't hear her yet because
We don't need volume.
Freddie.
But who's going to be the volume?
Ready?
Okay. All right, can you hear us, Daniela?
Yes. Can you hear me?
Yeah, we can hear you as well.
Okay.
Okay, all right, we're going to get started. I'm pretty excited about this talk today. We've learned a lot about insect brains, artificial intelligence, creativity, but this is probably the biggest view talk we're going to hear of the series. We're going to talk about evolution, life, the origin of multicellular organisms, the nervous system, and then we're going to talk about how that all applies to artificial intelligence. So backyard brains over the past few years has become a little bit better in our understanding of what neurons are and the difference in animals, and that is mostly due to our collaboration with Daniela Flores. We've been working together for about 5 years, teaching microscopy at high schools in Chile, and she just published a paper with us on a protocol to develop microscopes by directly melting glass. So she's a PhD student now at the University of Chile, doing wonderful research, but I want to yield the floor to her and allow us to learn what really is an animal and what really is a nervous system. So it's all yours, Daniela. Welcome.
Okay, thank you so much, Tim. I'm going to share my screen now. Please, you can, okay. You can help me to know if I actually sharing my screen, because I am not sure. Yeah, okay, thank you. Okay, that is okay? Is this okay? Okay, thank you so much. Okay, for the talk, I want to thanks to Backyard Brains to invite me here, and especially to Tim because, as he said, we have been working together for a couple of years and doing a very beautiful work in general in different topics. So it's very exciting to now to talk here about the origin of animals and their early evolution, and especially focused in nervous system because I don't feel like an expert in this field because I am working in my thesis is about morphological evolution in the transition dinosaurs to birds transition. So it's very different. But I like so much in general biology and in general in evolution in biology, but in the lights of evolution. So I work in the University of Chile in the laboratory of Ontogenia phylogeny, and also I want to show you this foundation because I developed with a couple of friends. I developed this foundation in Chile that is an interdisciplinary foundation, and we do a lot of dissemination of science also. So maybe, I don't know, maybe if you are interested or to know more about this, and maybe if you have some question after the talk, I put my email here so we can stay in contact, okay? Okay, so I'm going to speak in this talk about the origin and the early evolution of animals and the rise of the nervous system and its ancestral futures. Okay, this talk will be very wide in the topics that I'm going to talk, and but I'm trying to not speak a lot of specific things, but I'm trying to after this talk, hopefully we will have a wide thinking about how was the evolution of this interesting early divergence. Okay, so the talk contents will be first of all I will talk about a little bit of evolution. Then I will talk about early evolution of neural systems, and then Tim will join to this talk talking about what is intelligence. It's a very huge question, and then we will have time to speaking to conversation for the for questions. Okay, so a little bit of evolution. Obviously, I don't know. I am not sure what is your background exactly. Okay, so I prefer to put these images to begin. This image is an analogy between a clock and different important events during the evolution until our arise, okay, as a humans. So if we put in the last midnight, we can look the formation of Earth. And then we have a lot of time here, like until 4 o'clock or maybe until 2 p.m. Okay, of the other day. We can look that we have only here the origin of life, and we have all this time as a prokaryotic life. Okay, and then after this point, single-celled algae or single-cell organism, this in here approach sorry for my English, but maybe in 2 o'clock p.m. We start to look that begins eukaryotic life, and then sexual reproduction and different features that represent complexities in life. Okay, and only in 9 p.m. Okay, we can begin to look that the rise of animals, and then, for example, mammals, maybe 20 minutes before the last day, and only 2 minutes before we are as a humans here. So this is a good analogy to know how is the time of evolution, what we are speaking about. Okay, here we have another picture about how is evolution. Obviously, we have here the oldest microfossils. Very far, a long time, like 5 million years, 3,500 million years ago, and then first eukaryotes, and then ediacaran fauna. Here we have like the Cambrian explosion, and then obviously we are living here, but there's a lot. There are a lot of million years ago here. Okay, so
Daniela, can you hear me?
yes.
And as a scientist, if you could go in time, can you go to the slide before? Which of these epochs is the most interesting to you?
More interest for me? Wow, it's difficult to answer that thing, because I think that I am interested in all this area, all this period. But especially, I think that here, because during this time, maybe during Mesozoic, you have the divergence of the principle of the major dinosaurs, and that is more very exciting for me. And also, and birds are dinosaurs. And also during this time, because here we have the Cambrian explosion, so it's a very enigmatic period. We don't know so much about that, so it's very interesting. Okay, so I like so much this period, but also here because it's the origin of life, so it's more enigmatic also. But it's difficult to tell you what is more important for me.
Okay, thank you.
Thank you for your question. So specifically to this talk, I need to remember that animals in general, if you look this table represents the periods, the era or different periods that we that are developed by geological information. Okay, and using this geological information and also the fossils that are found them in different strata maybe, I don't know if but the different places that you found that fossils, and also with other techniques like molecular clock or biological techniques, it has been inferred that animals have around like before the Cambrian explosion, like around this time, okay, 800 million years ago, a lot of time. So and then during this time, it has been possible to find different fossils, okay, that represent the early evolution of animals. So when we speak about animals in terms of the phylogenetic view, of the phylogenetic tree reconstructions, we can see that here is a very synthetic phylogenetic tree, because we look bacteria, archaea, and eukaryotes. And eukaryotes now are it's known that developed from the nearest group from prokaryotic organisms are archaea, and bacteria are more ancestral. Okay, so eukaryotes obviously represent a kind of cells that it's more complex than prokaryotic cells. And in eukaryotic cells, in eukaryotic organisms in general, we can see that there is a lot of lineages, a lot of groups that evolve from an ancestral that here is not showed exactly, but they evolve in different groups. Okay, so when we speak about single-celled organism, eukaryotic organisms, we are speaking about a lot of organisms that are very different between them. Okay, here is another representation from that show how different groups like chromalveolates, excavate, euglenozoans, etc., that represents different groups of eukaryotes evolved, and the diversification or the different features that allow them to group together in different groups, obviously, are related to different steps of endosymbiosis. Because if you know, maybe you have been you have related to the term of endosymbiosis and the endosymbiotic theory of the rise of mitochondria and chloroplast. Yes? Okay, good. So in general, chloroplasts and mitochondria, when they are acquired and as an endosymbiont, they produce they can the possibility to produce different features in that organism, in that single cell organism, like and in this in different groups of unicellular eukaryotes, you can find different histories of primary and secondary and also tertiary endosymbiosis of plastids, and these plastids can generate other organs inside the cells. So these groups, these eukaryotic unicellular organisms are really exciting. They generate different features that are really exciting. But I will not talk about so much about this because I'm not. I don't want to talk about so much about this because it's a very huge topic. Maybe for another talk. So these different endosymbiosis generate the complexity of different eukaryotic unicellular organisms. Yeah, this is only a picture, only a couple of pictures that not represent the diversity, cannot represent the diversity of what I am talking about. Okay, but what interesting things is that during this early emergence and differentiation and bifurcation of these unicellular eukaryotes, there are some lineages that develop multicellularity. What is multicellularity? If you look here, you know that obviously in eukaryotes, we know that animals, fungi, and land plants have lineages of multicellularity organisms, okay? And there are some others, like fungi, that have lineages inside all the group that are unicellular, not only multicellular. So there are, for example, this image. I am not totally agree with all this characterization, but represent the idea. Okay. For example, land plants, all the organisms are multicellular. In the case maybe of slim molds, or we will talk about slim molds. Here represent all multicellular, but I'm not totally agree with this. But metazoans that represent animals, we are all multicellular, okay? And choanoflagellates that are the sister group of animal, we will see what is sister group, but is the an ancestral diversification that is near to animals. There are all unicellulars or some colonials that represent one step of multicellularity. Okay, that is what we can see here, that choanoflagellates is put nearest to the other animals. Okay, and as we can see here, sorry, I need to. Okay, as we can see here, we can look a choanoflagellate, a colony of choanoflagellates. Okay, so they have different possibilities to generate multicellularity, and that is very interesting. And other example of multicellularity that is facultative, is not an obligation, because in our case it's an obligation. We develop from an egg and a sperm that begins to generate a developmental process, and we all are not we all, but we in general, we as an animal are multicellular. But the slim molds is a phylum, is our organism from a phylum Amoebas, okay, and are represented, for example, by this organism named Dictyostelium discoideum, okay, and this Dictyostelium discoideum have the possibility to pass with different steps of behavior and of morphology, because they can stay like a only unicellular organism, and then they can aggregate, they can generate like a slug, and they can indeed produce this kind of structure that we can see here, named fruiting body. Okay, this occurs in general in condition of restriction of food. Okay, so sorry, what I want to show you is that it's more related to what Tim will talk about, is the similarity or the parallel between intelligence, behavior, and principally in those in two intelligence, behavior, and in my part of the talk I'm going to talk about neural system. Okay, but here I want to show you that it's an experiment that you can look a mass or a labyrinth that the researchers put cells of Dictyostelium discoideum, and they put cells here, okay, and here they put, after the labyrinth, they put like a food, okay, and they showed that the Dictyostelium can resolve, and then they can move to the food, resolving the labyrinth, and that is, we can see actually in this video also, okay, in this little video that showed how these slim molds can resolve the labyrinth, okay, and this is very interesting because we are talking about an structure and an organism that are living as a single cell, okay, and as a single cell they can comprehend or they can understand how is the structure of their environment to resolve their environment to eat. Okay, so
Can I ask you a question about these slime?
yes, of course.
So do they have difficulty digesting food or nutrients in like when they're isolated? And so like I'm just thinking if like 10% of the cells get to the nutrients, do they have a harder time digesting the nutrients than if say like 50% of the cells get to the food? Like do they digest as a community?
Could be. I am not sure. I am not sure of the answer of this question.
Yeah.
But what interesting thing is that in general, their conformation as a multicellular system or as a colony begins or arise when they don't have food. When they don't have food, they generate the configuration of multicellular system. So
That's fascinating.
probably is related to the amount of food. Obviously, is a relation there between the amount of food and the configuration as a unicellular organism or a multicellular organism. And that is interesting also because it's not only this relation between food, the presence of food, and the configuration as a multicellular organism is related to this organism, to this Dictyostelium discoideum, as example, because in choanoflagellates, that are the sister group of all animals, they are unicellular, but they can develop multicellularity as a colony. And one group in Berkeley, they showed that when they put a bacteria that is a food of choanoflagellates, they acquire the multicellular configuration. So probably there is a relation between the multicellularity, the unicellularity configuration, and obviously the other food or other maybe oxygen, other stimuli that are around the system. I am not sure about your question, but maybe if you want, we can. If you write me an email after, I can I don't know, write more, read more about that. Okay.
Yeah, I'm just interested in the connection between the processing as a system and the processing as an individual cell, like similar to the way that neurons process things individually and then also as a collective. But thank you. It was a great answer. You can go on.
Okay. No, it's an incredible topic. So okay, maybe later. So what I wanted to tell you also is that here this behavior is not only related to an imposed gradient. It's not only related to put a food, but also this behavior in the case of Dictyostelium discoideum, this interesting behavior is related more to a self-generated gradient. That means that when they find a gradient of food here, a gradient of, I don't know, some molecules that they sense, they have the chance to degrade this molecule and then to move more than if they don't have the chance to degrade the food or other molecules. Okay, maybe this is not totally interesting for this talk because this is more related to behavior, but also to intelligence. But this kind of behavior we can look, as Samuel said, we can look this kind of behavior of not only in this unicellular organism, but also in developmental biology. Also, when one cells move between a developmental system. So it's very interesting. Okay, so from this a very big ocean of protists or unicellular eukaryotes, we can these different characteristics of or features that have these eukaryotes, we can find and we can use these characteristics to develop a phylogenetic tree. And obviously we know that animals, here we can look animals because we have here the early diversification of animals: Cnetophora, Porifera, Placozoa, Cnidaria, and Bilateria. In Bilateria represent the big group that groups different, very different animals. Okay, and we will speak more about that after. But in this talk, I want that you remember this representation, that here before Bilateria diversification, we have these four groups: Cnidaria, Placozoa, Porifera, and Cnetophora, that are the first diversification of animals. And for that reason, here in these four groups, we can find more basic, or maybe some features that represent transitions between our ancestry as a unicellular organism to this organism Bilateria. Okay, so we will find different features that are very that represent that transition. Here, yes, sorry.
Okay. Just I wanted to clarify for those in audience that do not have a biological background. Bilateral is almost everything. Cnidaria is the jellyfish and sea anemones. Placozoa is like this weird bonnet thing in the ocean. Porifera is a sponge, and Cnetophora is like this other kind of weird animal that moves with flagella. So anyway.
Yeah. Sorry. If you want some question about one groups or whatever, just tell me, please, because sometimes I forget that I am a biologist and I always look this kind of names, so I forgot that things. Okay, so when we speak about this fierce diversification, we are here. This diversification of the first animal occur during this time. We don't have, during this time, we refer first to the Cambrian explosion, okay? So, first or more than the Cambrian explosion that occurs around 500 million years ago, okay? In the Ediacaran period. Okay, this is a very different topic, but I have 2 slides to explain this, but only to have to speak all we can. I want that all we have the same background to the rest of the talk. So, respect to the phylogenetic reconstruction, because we can see here a phylogenetic tree, but we don't know how this phylogenetic tree is developed, okay? So when we look at phylogenetic tree, this phylogenetic tree represents how animals, in this case, or how groups of organisms share some features and don't share other features with other, okay? It's a kind of classification. So to do this kind of classification, we use the morphological approach as this kind of approach when you compare morphological structures, like here the forearm, okay, in the case of tetrapoda. Or I'm sorry for the Spanish, I didn't remember to change, like a ancestral tetrapoda. But also we can use here in the case of fossils, obviously you only have the morphological approach because you don't have the molecular. Sometimes they describe some study describe that they found proteins or other things, but it's very difficult, are very degraded. Okay, so but in the case of extant animals, we can use, for example, a molecular approach also. Okay, using not only this represents a sequence of nucleotides. Okay, a sequence of nucleotides. Okay, but also this is possible to do with proteins. Okay, but this represents a molecular approach. So when we describe a phylogenetic tree constructed to describe metazoa, in general, we used not only the morphological approach, but also the molecular approach. Okay. Another thing that is important to notice is that when we speak in terms of evolution, we are speaking about groups. Okay. Maybe if I refer to the C group, I can describe. I know by this phylogenetic tree that C is more related to B than to E. Okay, and in general, we describe B in the case of C as a sister group. If I say that I am studying this group, all this group, from this node to all this group A, B, C, we refer here to the in group, and the other group that is nearest to that in group we refer as a sister group. Okay, that is important because we know that, for example, if this is all animals, metazoa, we refer as a sister group to a choanoflagellates, the choanoflagellates. Okay, so and other thing that is important to notice is that when we speak about all this group, the futures of all these groups, we refer to monophyly. What is that? It's a natural group. When we refer as mammals is a monophyletic group. Okay, because it's a natural group, because have the ancestor and all these descendants. Okay, in the case of Pisces, okay, we know that Pisces is not a phylogenetic group. It's not a monophyletic group, because if we say that Pisces is a monophyletic group, we need to speak that Pisces, a group, also all those descendants. Okay, but in this case, Pisces represent a paraphyletic group because not they don't group all the descendants. Okay, but what I am interested to that you remember is the name of monophyletic group as a natural group. Okay, the other is a little bit functional groups, are not natural group, but functional for our narrative, like a biologist. In this case also, other thing that this is important is that between nodes, okay, here we can represent the innovations, the evolutionary innovations. Here, for example, this is the amniotic egg. Okay, so here some ancestral groups, some ancestral organisms develop the amniotic egg, and then the amniotic egg was a innovation of all this group. Okay, I put also this picture. I don't know if you know what picture is. Somebody can imagine from what?
Charles Darwin.
Yeah,
Yes.
Darwin and origins.
Yes, it's like the first phylogenetic tree. So it's a very important image. Okay. So why I'm talking about phylogenetic trees is because there are some groups in the case of animals that are very robust. Depending of the morphological information or molecular information, they are robust, they don't change. But in the case of the first organism that evolved here, the first organism like sponges or Netophora, this kind of similar medusas, sorry, the relation between those group are not totally clear. So there are three hypotheses that put, for example, in one case, one phylogenetic tree put sponges like a sister group of all other animals. Okay, the sponges was the first animal that arise in the evolutionary history. Okay, that explains this hypothesis. The other hypothesis is that Netophora. Okay, we will speak about what Netophora is exactly, but Netophora is a sister group of all other animals. The other hypothesis is that sponges and Netophora are grouping together, and this group is a sister group of all animals. So this what is important to notice is that these different phylogenetic hypotheses represent different. We can use these different hypotheses to understand different scenarios of evolution. Okay, we will speak up more about that. But for example, as a spoiler, Netophora have neurons. Okay, but sponges doesn't have neurons. So here the scenario is different, because if sponges don't have neurons and then Netophora, yes, probably the rise of neurons will be here. Okay, but in the case of Netophora, have neurons and have a very basic but have a neural system, and Porifera doesn't. So probably Porifera lost all these features. So the different hypotheses are very important to understand how evolution proceeds. Okay, and the other thing that is important is that when we speak about Urmetazoan, what is Urmetazoan? Is an hypothetical ancestor of all other animals. Okay, so here the Urmetazoan is an hypothetical ancestor of the Metazoa in general. So we can see that choanoflagellate, sponges, Netophora, Placozoa, Cnidaria, or Bilateria, okay, have different features. For example, here sponges have multicellularity, have collar cells, have oogamy, have eggs and sperms, okay, have epithelia, have gastrulation, that is the first movement during the developmental process. But in the case of choanoflagellate doesn't. So this when we want to think how the Urmetazoan was, is important a phylogenetic tree, because we know that here the first diverging groups in this first evolution of animal, they have different characteristics. Okay, so we will moving on to the other topic, that is early evolution of neural system. We know in general, I'm going to try to speak a little bit about different characteristics of these early groups, okay? But I don't want to be specific. So sometimes I'm going to show some images, but I will not refer to a specific molecules or something that appear in the images, but only the general view. Okay. So in general, we know that neuronal types or neural system in general proceed hypothetically from a first neural cell types that sometimes some researcher like this researcher think that probably derive from a choanocytes. Sorry, Tim, can you?
Yeah, that's what the hypothesis is, that the first neuron could be derived from
What is the word exactly? Derived. Yeah, sorry. Derived from a choanocyte or mesenchymal cells. And then after the evolution, they begin to generate a nerve net composed of neurons that we can see this kind of structure, this kind of organization in Cnidaria, in Medusozoa or anemones, and in comb jellyfish, okay? And after this represents more what happened in Bilateria. In Bilateria in general, we can find a centralized nervous system, okay? That obviously we know more about this kind of system here. So, in general, this picture is very big, but I am not talking about all the what I want to show you is that in general, Placozoa here we have the early diversification. We have here one branch that represents choanoflagellates, and here one early diversification. In this phylogenetic tree, they put Netophora as the first branch of diversification. And Netophora have neurons and have nerve nets, okay? And also they have muscles, okay? And a kind of the mesoderm, okay? So, but Porifera, sponges, they don't have neurons and muscles. The same occur for Placozoa, okay? They don't have neurons or muscles, and in Cnidaria, representing by anemones or medusas, they have nerve nets, and in the case of all other animals, in general, here we can find central nervous system, and in some cases, in some lineages here, obviously some lineages lose some complex characteristics, and they have nerve nets, for example, or other configuration. But in general, here we can find a more complex and a centralized nervous system. Okay. In the case of what happened about the early evolution of neural types, what is interesting to notice is that when we speak about a neuron, okay, we are speaking about a morphological. We are speaking about a lot of things related to what is a neuron, okay? It's not only a morphological point of view of a neuron. We know that neurons have dendrites, have an axon, have different morphological features, and a functional features also related to this morphology. But also we have we can relate the identity of a neuron about from a molecular viewpoint. What kind of genes have, what kind of channels have, etc. Okay, and one important things about neurons is that neurons obviously have this capacity to develop action potential. Okay, so but this kind of phenomena depends about voltage gated channels. Okay, and what is interesting to know is that different groups like Netophora, Comgelis, or in the case of Cnidaria, and then after diversification of Bilateria, we can find different voltage gauge channels. Okay, in this case, for example, in Netophora we can find a potassium voltage channel. In the case of Cnidaria, more kind of voltage channels like sodium, calcium, potassium, and here we have also different configuration, different acquisition of these genes and these functionalities. So, but what is interesting to know is that here we have some characteristic of voltage channels in Netophora and in Cnidaria. These both groups have neurons, have neural systems, and in the case of Placozoa and Porifera, that they don't. But this kind of voltage channels, ion channels, what is interesting to notice is that they arise before the generation, before the arise of animals. Because some groups of protozoa or protists, they have this kind of voltage channels. They use these voltage channels for other things. Maybe they don't have a conventional synapses, or they don't have action potentials, okay? But they use these channels for other physiological processes in their lives, okay? But they have some groups, obviously not all groups, but we can find some groups of protista, unicellular eukaryotes, that have this kind of potential channels, and also other things that are important, like receptors of some neurotransmitters like glutamate. Okay.
I have a question. Yes. That's right. So one of the people here at Backup Brains is working with like plants and trying to see like signals in plants, electrical signals in plants. Could you comment on, are there like say sodium voltage gated channels in plants, or can you say anything about what voltage gated channels might exist in plants? Maybe a tomato plant specifically?
Okay. That is something I think that Tim will speak a little bit.
Okay.
I am not sure, Tim.
Sure.
Okay. Okay, but I am not. I don't know so much about what is the specific process, or what is the specific molecules involved in the plant, in the plant's action potential, because I know that obviously plants have action potential. Actually, the first synapse, I think that was known. Okay, I think that was discovered in plants. Okay, but I am not sure, because plants are very different. It's a very different system. It's a very different system, and they also, but probably Tim will speak about that, because they move, but with the flux of water and other things. So it's a little bit different, but I am not sure. Okay, I am not sure what are the process. But what I want to tell you with this image is that voltage channels that previously was in general was thought that only animals has this kind of genes, that's this kind of proteins related to the synaptic process. But it's not the actual the current scenario. Okay, because when we explore all this ocean of protozoa, we can find a lot of features that they share with animals. So they have they have the grains of the sun to generate the beach, something like that. It's very beautiful, I think. That is because I think that is very beautiful, this to know more about this protist. Okay, so this obviously here there is not a lot of information about this, because this is a field that is not that only for a few years ago begins to develop more. Okay, but we know that neural system. Okay, we know more about neural system not only by a molecular viewpoint, obviously, but also with the morphological viewpoint. As I told you, using fossils, for example, I don't know if you know or if you listened about the Burgess Shale place that is in Canada, where some researchers find a lot of organisms that represent this Cambrian explosion. Okay, and here when we see fossils, here we are seeing a fossil, and here is another. Here is an insect that they compress and desiccate to looks or seems like a fossil, and they can compare the neural system of this insect to the fossils of some of the organism that it's possible to find in this Burgess Shale place. Okay, so in the fossil, it's possible to look how the neural system also here a centralized nervous system arise. Here we can find in another fossils, Ottonia prolifica, another description of neural system, and in the case, for example, with these two organisms, obviously are fossil organisms, Waptia and Fuchsia unguaya, we can look like a head, a centralized nervous system that it's not a nervous system, obviously, because this is rock, okay, but represent the same attributes, the same features as other organism that we can look now, extant organism. Okay, so we can see we can understand a little bit more of how was the evolution of neural system, also about using fossils. Okay, so
can I make a comment on that, Daniela?
Yes, of course.
Yeah, the Fushan-Huya fossil is used to like show what the progenitor, what kind of like cerebral structure they had in the optic lobe. So there's this little arrow to the OPT, and so they were like showing that the Fushan-Huya has like similar structures with the medulla and the lobula and lamina, like insects have in their visual lobes nowadays. So that is already kind of like a preserved structure to build a visual system like that.
Yeah, thank you so much. Yeah, it's incredible. I think that these images are really cool, really beautiful, because the preservation of this structure is very good. It's very difficult, the preservation of the structures in general in animals, and in this early diversification, because there are some animals that are soft. This soft structure is very difficult to preserve. Okay, so yeah, it's really beautiful. Okay, moving on. Now I'm going to speak a little bit more about these groups specifically as sponges, Placozoa. Placozoa is only 2 species, but is a very simple animal. They have only 4 or 6 kind of cells, but they have only 3 layers. Yeah, okay. In the case of sponges, they are very simple also. Cnidaria have anemones, have medusas, have hydra, and corals also. And this is Cnidaria, and also this early diversification have the Netophora group, and Netophora is like something similar to medusas, but they have other things. They have different organelles comparing to Cnidarians. They have coloblasts. Cnidarians have cnidocytes, or they have this kind of line of cilia. They have other characteristics that they put their group together as another group. Okay. Sorry. So what is interesting also is that this early diversification of neural system is difficult to speak about, because we know that we are an organism. Okay. It's obviously, when we speak about evolution, when we speak about, I don't know, the eye or an arm, okay, obviously that feature is inserted in a system. So it's difficult to speak about neural system without sensory structure or without muscles, because neural system obviously have a function, promote a function, generate a function in the system in general. So I wanted to highlight that in the early evolution of animal, there are also sensory structure in basal metazoa. Okay, there are a lot of sensory structure. I put here only different structures, but only a few. Okay, here, for example, here this image represent is an immunological labeling of this label that represent cells that are related to neurons. Okay, so this FMRF represent cells that are similar to neurons. In the case of the planula larva is the first stage after embryogenesis in Cnidaria, and also this mark we can see in the tentacles. Okay, and we know that Cnidarian in general are very responsive to the mechanical stimuli of the environment. Okay, for that reason, maybe if one time, I don't know, you are swimming in the beach, and then you look at a medusa, and then you feel you feel sorry, a stick, maybe a stick. Okay, it's because they have this kind of stingy cells, stinging organelles named cnidocytes, that they have actually like an harpoon, like a sting. Okay, that for mechanical stimuli here in the cnidocyl, that is like a hair, they promote the expulsion of this kind of arm. Okay, this kind of sting organelle. So this is a kind of sensory structure in the case of Cnidaria. In the case of Porifera, for example, it had been thought that also a kind of cell that is in general in all sponges, named choanocyte. Okay, because and is very very similar also to the choanoflagellates that are the sister group of all animals. This kind of cells, it's supposed to fill for this flagellum, and using this microvillus also is used also as a sensory structure, okay? But these are only a few examples, okay? Because in this early evolution also there are other sensory structure or sensitive structure.
Daniela?
Yeah.
Maybe in the next 5 minutes, try to wrap it up so we can talk a little bit about intelligence and wrap the talk up. So.
Okay. So I'm going to move on. What I wanted to show you here is that in general, the evolution of synapses also depends about different molecules that are implied in the evolution of synapses. And as I said before, there are a lot of molecules that diverge in eukaryotes before metazoans, okay? And the same occur when we speak about the different molecules that are involved in the postsynaptic structures, okay? Here we can see that a little representation of postsynaptic components, and that begin to be more complex from choanoflagellates, porifera, sponges, anemones, and insects, etc. Okay. So, in the case of Placozoa, when we speak about Placozoa, this kind of organisms that are very, very basic, we can find also neurosecretory cells. Placozoa doesn't have muscles, doesn't have neurons, but they have this kind of gland cells that, interestingly, these gland cells marks for some molecules that represent the identity of neurons, okay? So despite they don't have neurons, they have some marks related to neurons, and they have some genes related to neurons also, okay? And they have also other kind of cells named fiber cells that we can see here, fiber cells, that is interesting because this kind of fiber cells contact with other fiber cells, and this contact is reminiscent to the synapses, okay? But they don't have neurons. In the case of porifera sponges, they don't have neurons also. They don't have muscles also, okay? But they have a behavior that is very, very interesting, because despite we cannot imagine that sponges have a behavior because they are sessile, they are they don't move, okay? Sponges have this kind of behavior of contraction and expansion, okay? And this is very interesting because the different lineages, the different groups in sponges, okay, different organisms in these big groups of sponges have this kind of possibility to contract, okay? And this is depends, for example, of external stimuli like mechanical stimuli that induce contraction, but also they have this kind of contraction and expansion in a rhythm during a period of time. Okay, so this is important. What I wanted to show you this kind of behavior of porifera because they don't have muscles, but they have a behavior related to muscles. They are a similar behavior of muscles to muscles. Okay, and also porifera sponges have also different neurotransmitter. Okay, for example, GABA. Okay, that we know that GABA, another neurotransmitter, we use this kind of neurotransmitter, but in the case of porifera, they have this neurotransmitter and they have the genes for this neurotransmitter, but we don't know what is the what is the function of this kind of neurotransmitter. Actually, they are 3 neurotransmitter: GABA, I don't remember the other, acetylcholine, but there are few neurotransmitters that it's supposed that induce the contraction, and other that induce relaxation of the system. So it's interesting because in here neurotransmitters are involved in the behavior of the sponge. In the case of Cnidarian, in the case of anemones, for example, we know that Cnidarian have a very simple but different, they can pass with different cycles of life. And in the case of the adult, in the polyp, we can see here neurons label. Okay, in the case of Cnidarian, all their body have neurons and have this nerve net. Okay, and here we can look that from a blastula, from a gastrula, from phases of the embryology, they begin to generate neurons, and then in the planula, these neurons begin to expand after they generate this more complex configuration of the nerve nets. Okay, and here we look neurons, but also here this image show that not only we have the same kind of neurons, but also we have different kind of neuron in different position. Okay, because they mark for different neurotransmitter or etc. Okay, moving on. I don't want to speak about this. I don't want to speak about this also. This is only to represent that different molecules that are involved in bilateria neural system are also present in the Cnidaria, and in the stem cell, and in the neural precursor, and in the neural generation in Cnidaria. Okay, and okay, I'm moving on. The only group that I left is Cnetophora. Cnetophora is a very, very, very interesting group, because we know that Cnetophora, if we look here, is very, very complex. Actually, they are not so complex like bilateria, like insect, like worms, like vertebrates, but they have different structure, like neural nets, like ciliated combs that are representing movement of this organism, and this kind of organelle, the coloblast, that are sticky organelles. Okay, they stick to a prey for their consumption. So and what is interesting to notice is that we talked about that Cnetophora. We don't know how Cnetophora is exactly, because this group Cnetophora is more complex than Porifera and Placozoa, but some phylogenies put Cnetophora at the beginning of the phylogeny of the animals, or other phylogenies put Cnetophora like here in the position of Porifera, or other as a sister group of all bilateria. So it's interesting because Cnetophora have neurons, have muscles, but here Porifera and Placozoa maybe lose them. Okay, Cnetophora is more complex because they have an avoid sensory organ. Actually, this sensory organ, we can look here, is like a basic brain. Some researchers describe that Cnetophora have a reminiscent or have a basic centralized system, okay, neural system. And in the rest of the body, they have a nerve net configuration, okay? And what other thing important is that when you look the synapses of Cnetophora, okay, you can look the same configuration or a configuration very, very similar to the vertebrate synapses. Okay, here we can see vesicles, we can see a mitochondria, we can see a very near functionalities with the smooth endoplasmic reticulum, and it's the same. It's very similar what we happen in vertebrates, and when they identify all the molecules involved in this synaptic process in Netophora, it could be described that all these molecules in red and in orange are conserved to the other synapses in all animals. Okay. If you remember, I told you a little bit about fiber cells in Placozoa. And here is a representation of how fiber cells here actually have a very, very near contact as a synapses compared to synapses in other groups. Okay? Well, no. Okay, finally, I want to speak a little bit because this Netophora, the Netophora position in the phylogenetic tree is important because if we know that Netophora, if you imagine that Netophora have nerve nets, okay, it's possible a multiple origin of neurons. Here in Netophora, and then a loss of neurons or a primary absence in Porifera and Placozoa, and then a reacquisition of the signaling of the genes of all of this related to the neurons. And the other scenario is to think about a monophily origin of the neural system of the neurons. Because if we know that a single origin of neurons and their neurotransmitter and their signaling, etc., implies that Netophora have new neural genes and transmitter. They acquire independently a neuronal genes, and then Porifera and Placozoa, they don't have neurons, they lost the nervous system and transmitter, okay? They lost here in Placozoa also, and then in Cnidaria and Bilateria, they reacquire this. So this two scenario is very important because the loss of structure, the loss of genes, the loss of functionalities, and the reacquisition of that is very difficult in evolution. So this scenario, I put this last slide to explain that it's important to clarify how is this early phylogenetic tree of bifurcation of early animals to understand or to hypothesize about how was the origin of neural system and obviously the futures of neural system. So Tim, now is your turn. I'm sorry for the delay.
It's okay. All right, so I'm going to go pretty quickly, but how do I present? Okay, I'll just use my screen.
Okay.
Sorry. All right. Okay. Share. Okay. So animals evolved nervous system tissues, and that was a very good introduction. I know there's a lot of biological terms. I find it fascinating. We can say that for a week. But animals evolved nerve systems to perceive the world and respond quickly. So to sense and move. That's why we have nervous systems. But as a result, we have gained intelligence. We tried to gain intelligence. As kind of a benefit of having a centralized nervous system. So what is intelligence? And this is like one of those philosophical terms that we could run a 10-week short course on where we just argue about what intelligence is, and then I'll try to write a paper at the end. So this is one of those unresolved philosophical questions that will be discussed for the next couple thousand years. So I just made this up. So Daniela and I just talking made this up. So there's no like background where we came. I was just thinking about it. So how do we consider a person to be smart? Like that person is intelligent. When that person is intelligent, that system is intelligent, it has knowledge. I'll explain that, right? And then also, if they have flexible behaviors. If you can change, if you can learn, then you're probably smart. I like Etienne, but if Etienne was exactly the same as when I first met him 10 years ago, we wouldn't. You should change as you experience life. So kind of if we consider an intelligent person someone who grows as their life progresses. And then also there's another requirement for intelligence is a theory of mind, which is what children get when they're 3 years old. And so again, we're talking about artificial intelligence. We're talking about what is intelligence. This is what Tim thinks intelligence is. If you ask me, do I think I'm intelligent? If you ask me, do I think that other person is intelligent? These are probably like the four things, I would think before saying yes or no. Okay, so knowledge in a human. Naturliche Wissenschaften. That means nature in German, right? So we have the knowledge, right? So as humans have knowledge, right? Schmetterling is butterfly in German, right? All right. But also metazoans are broadly animals. We all know this cat. This is Benji. It's an indoor-outdoor cat. So the cat goes out, maintains balance the bunny population in Ann Arbor, and then it comes back home. So how does it know where its home is, right? Maybe smell, other types of cues. So this animal, it doesn't know that Naturliche Wissenschaften means nature in German, but it does have knowledge. So like the system does have information. So that's one measure of intelligence. All right, so this is Alejandro, Alexander the Great. And what's he doing? Does anybody know what he's doing in this picture? Whoever could cut this knot,
cutting the knot,
rule the world, the Gordian knot.
Yeah, cutting the knot.
It was very, very tightly knotted up. He just took his sword out and cut it. That's kind of behavioral flexibility, right? So behavioral flexibility could be, learning a language, learning mathematics, learning how to code Python, learning how to use Colab. But these are behaviors of yours that you change and evolve as you go through your days. And we consider like someone not very intelligent if they never change their behaviors, right? Or like that famous wasp that if you take the cockroach out of its burrow, it comes out of the burrow and brings the cockroach back in. And you take the cockroach out, it comes out of its burrow, brings the cockroach back in. It never realizes that, hey, something's up. It's like a machine. We're all machines at some point. Okay, and metazoans or animals have a behavioral adaptability, especially the corvids, the ravens, the blue jays. They can learn how to use tools. They can learn by observing. They can solve problems. This is a kind of a small but very exciting world of animal intelligence. Again, since we do a lot of education work, I try to tell students that you should constantly be trying to progress as you go through different phases of your life. So humans age, we gain experience, we have the experience to our personality, and hopefully become more enlightened people as we go through life, and not less enlightened. And this is kind of harder to state the case with in other metazoans, but there are complex societies like in elephants, in whales, and other non-human primates where the boss can no longer become the boss, or the societies, the role that each person places in that little society changes as they develop, from babies to seniors. Does anybody know what the theory of mind is? Anybody chime in on that?
Is that just basically the understanding that you yourself are thinking, or that the understanding that other organisms also are thinking?
What are other people think? What are other people thinking? This is also empathy. So sometimes when I'm really mad about who someone can't understand me, I'll say that person that hasn't developed a theory of mind yet. Like they don't know how I feel, right? Given my situation. So the classic thing is we have I have my gummy bears here. I have my gummy bears in a box. Everybody see the gummy bears in a box?
Yep.
And I leave the room, and then Etienne puts the gummy bears in my book bag, and I come back. Where am I going to look for the gummy bears?
In the
The box.
In the box, because I don't know that Etienne put it in my book bag. And so if you ask a 3-year-old, that they're going to say Tim is going to look in the book bag because that's where the gummy bears are, right? But the 3-year-old isn't capable of saying, well, I was in the room with Etienne, but Tim wasn't. And therefore, if I were Tim and I wanted gummy bears, I'll look in the box. Does everybody understand that? Okay, so that develops in people around 3. There is some evidence that animals have theory of mind because types of corvids, when they hide their food, they don't want other people to see them. So I mean, that's kind of, maybe they just don't want people to see them. Not thinking like, well, if I was that raven and he saw where I put my food, you know, like. So, but there is some. This one is one of the more controversial. But this is other metazoans could possibly have theory of mind. Okay. So we'll talk about nerve systems and complex nervous systems, but you know we have had like, are we being metazoanist? Because intelligence exists in other systems. So Etienne and I are studying plants and plant signaling. Also, there's some evidence that plants could potentially learn to associate wind with the light source. And not many has been published on this, but in the recent edition of American Biological Teacher, there was a scientist named E.T.M. Serb that published a paper on this.
Never heard of him.
Who's that guy? Oh, weirdo. Okay. And then Daniela talked about dictostellium. This is that cool transition between unicellularity and multicellularity. So in a dictostellium, it's an amoeba and it runs out of food. It'll form these colonies and then turn into these flowering bodies that then burst, and then the spores can travel. The amoeba can travel farther. So the individual amoebas don't have food. They all, quote unquote, make the decision to work together to save. You know, and the amoeba that make up the stalk will die, but it's for the greater good of the amoeba individual, which is only individual one. It's weird, right? Okay, it's weird. All right, you all know this. Okay, so but now this fellowship is the machine learning and neuroscience fellowship. So we're doing machine learning on mini computers or microcontrollers. So. And can a microcontroller be intelligent? You know, what is an artificial intelligence? What does that mean for an electronic adding device? What does it mean for the nanosense BLE to be intelligent? We all know what computers are. They're computational machines written in language. You all know this, right? So this is not really important. You know, we all know what an artificial intelligence is with artificial neural networks and nodes. We've been studying that for 9 weeks now. Okay, this is kind of like I know we're running late and it's kind of a little hot in here, but Daniela and I did want to open up maybe 5 minutes for discussion to see how you all feel, because I just feel like the question of intelligence in machine, like what does it even mean? You know, so intelligence in computers, does it have knowledge? Does it have behavioral adaptability? Does a computer progress through change as it ages? The computer had a theory of mind. And I guess we could program systems that would have these characteristics, but is there a fundamental difference between metazoan intelligence and electronic adding machine intelligence? And I kind of want to open it up to that. So does anybody have anything they want to add?
I could add something on that. I think that a lot of the way that intelligence gets labeled in terms of like when we study in biology is based on whether or not something is beneficial to the organism. But I think in general, like we can attach a lot of like what we call intelligence to just the complex functioning of information like together in systems. So I think that when we apply it specifically in the case of artificial intelligence, I think the thing that makes it most similar to biological intelligence is just this idea that we start to specify motivations. We start to specify the idea of achieving goals through the connection of information to outcomes.
So an information processing system that desires to achieve goals for itself as an organism would then be an intelligent entity.
Or at least to say that when there's no motivation that's clearly defined in a system, then it's hard to assess intelligence because there's no measure of whether or not something's doing better or worse. So I think that the motivation is a really important part of intelligence.
Good point. I like that.
I would like to point out that I said intention in the chat before Sam spoke. So
intention and motivation. Okay. Anybody else want to pipe in?
I don't know, the thing that's most curious to me here is where are we really drawing that line of defining motivation as a key part of this process? Because, you know, babies inherently just kind of exist, for example. You know, they just kind of toddle along. And a lot of their experience is attempting to learn things via what sensory things they have, you know, shoving things in their faces for the most part. You know, and that's not necessarily motivated, at least in a way that we might be able to see it. You know, it might be a general curiosity, but where does, like, how are we defining that in the context of a artificial intelligence?
Well, according to the Marzullo or Flores-Marzullo theory of intelligence, a baby's not smart until they're 3, because that's when we get the theory of mind. But that's just making a hard line, you know. Sometimes you play with 2-year-olds and you get the sense that they're smart, but they don't, you know, why they're smart. Usually because they seem to be learning pretty quickly, you know. Yes.
Sorry. I was thinking about this question, but where do we draw the line of defining motivation? Because I think that motivation, yes, is very, very important to define intelligence, but motivation of what? Because if you think that one organism, whatever organism that you are talking about, maybe a bacteria, they have the motivation to live. They have the motivation to survive. They have the motivation to eat. So maybe it's difficult to explain. Maybe all organisms in that sense are intelligent, but there are different complexity of intelligence. I don't know, because all organisms have a motivation. Of what? Motivation.
This is something that I don't necessarily have, like, I can't really speak on this from a scientific perspective, but just in some of like the like Buddhist like thinking that I've like paid attention to a little bit, there is this concept that intelligence is sort of in all things. But I think that the way to sort of connect that back to what we were talking about here is really that any entity that's able to sort of have a clear boundary between itself and something else, it has informational structure, and then that informational structure may be well adapted or poorly adapted to the circumstances it finds itself in. And so really the only thing that's capable of having a motivation is something that has some clear boundaries. So I guess to a certain extent you could say something expresses intelligence when it increases its ability to stay as an entity itself, and it uses the information at its disposal in a way that increases the likelihood of it propagating itself, like survival, basically.
Yes, it's very interesting what you said, because there is a study. There is a different study also, but I have read one of them that relates the intelligence, the generation of the rise of intelligence with the immune system, because immune system, what is the function? They describe that the major or the first function of the immune system was to generate identity, because they use early organism and ours also, and us also. We use the immune system to recognize our system from the others. So it's interesting to not only to refer to the intelligence, referring to the neural system, I think, but also to the system in general, as you said, related to the identity.
Yeah, and so in that same way, I know somebody in the chat mentioned parasites. I think that like then you could maybe get a clearer definition of intelligence as a beneficial structure of connecting sensory input to some sort of motor output.
Yes. I really agree with you.
All right, so Daniela has agreed to hang around for 10-15 minutes after the talk. For those of you that still want to hang out and continue this conversation. I think it's a fascinating topic. You know, think about the beginning of the talk with talking about the big question of evolution, and where are we now with this machine artificial intelligence revolution that we seem to be enjoying. So, Daniela, thank you for taking time out of your busy doctorate schedule to present to us. And anyone who wants to continue talking to her, either online or offline, you can see her contact information there. So, thank you, Daniela.
Thank you very much.
All right, so we're going to hang out for a bit if anybody wants to continue the conversation. But I know the internship is ending soon, so some of you want to get back to the code. So.
Does it end soon?
Not in your heart. In your heart, you can.
Let anybody else ask their questions first if they want to.
Go ahead, Sam.
So I just wanted to, I guess, talk a little bit more about some like organisms that have specific, maybe like, I'm obviously like this is all centered around the development of neurons and the way that they connect into larger systems. I was wondering if maybe we could like pick an organism that maybe is at like an interesting point along that developmental pathway to talk about a bit. I know that during the talk you mentioned that I can't remember the name of what it was, but it had a neural net as well as a centralized nervous system. What was
Metaphora.
Netophora. Yes.
What can you say that one more time?
Netophora.
Netophora. I'm just, I guess, like, first of all, I was curious about like how old that is, and then also where that doesn't fall in like our lineage, correct? That's a parallel line of development, correct?
Okay, Netophora is really old. We don't know actually when was the rise of Netophora, because we don't know is the exact position, if it's at the beginning of animals or is more as a sister group of all bilateral animals. Okay, but definitely. But what is important is that Netophora arose during the Ediacaran period before the Cambrian explosion. So we are talking about 600 million years ago, approximately.
I guess another. So like when I was really fascinated to hear that there's any organism that has both of these features, and I guess one of the things that it makes me think about is the possibility of having some like Descartes referred to it as like the seat of the soul in the brain, like having some like location that sort of is the center of consciousness or something like that. So I was wondering if like in these very early sort of centralized neural systems, if there tend to be something like an individual controlling cell, like that would be like similar to like the seat of consciousness in the human brain. I don't know if you could comment on that.
Okay. I think that to speak about conscientious is very difficult.
Yeah.
And obviously here in these early animals, because they are not a centralized structure. Actually, Netophora has this aboral sensitive organ, but it's not a brain. It's not a brain. So in general, these early animals like Netophora or Cnidarian, they have nerve nets, and they use this neural network only to move, to contract muscles around these neurons. Okay, but I don't know. To speak about conscientious is very difficult, because it's like for me now it's a little bit metaphysical. It's not. Yeah. It's very very interesting, but I don't know. I don't have them. Actually, there are a little studies of Netophora of these early branches. Okay, it's not. There are not a lot of studies, and there are any study. I haven't looked any article speaking about consciousness. So I don't have the I cannot feel well speaking about conscientious because it's not something that I have read about.
And there's a huge debate. There's a huge fight in the community as to whether metaphors evolve neurons independently or not. And so papers come out every year like, no, this is why we're right, and this is why we're no, this is why you're wrong. It's kind of actually kind of cool. It's pretty.
So just a question here, like what's the difference between decentralized or systems versus just neural nets? Like what makes a difference besides this distance between neurons? Do we have any specialized structures that arise?
I think organization mostly, that the other one is diffuse and ours is more structured, layered.
Yeah, ganglia. This is like a ganglia of gummies, right? So just like put the gummies on different parts of the table. It's a gummy net.
And for the consciousness question, wouldn't that be a lesion or a stroke if there was just like a single neuron or a single area in the brain? Then we need to have kind of just per like coincidence people to lose only their consciousness because they lost the functioning of a little part of the brain. So I think it might be difficult to pinpoint it down to a single area or even a single neuron.
And also, in one of the projects that Daniela and I have worked on is that I think I talked about in the first week was the anemones that this marine invertebrate electrophysiology, like with the medusas or jellyfish, and then the anemones is very not much is actually studied about how nerve nets work. They're hard to record from. It's like the plant stuff is just so small and it's wide open. So we actually tried to catch anemones off the coast of Chile and record from their nervous system, but we never got anything but noise. But we'll get it one day. Yeah.
Do you know if anybody is doing anything like
We are trying. We are trying.
Oh, good. There's some where you can cut a tentacle and put it in a suction electrode, and as the tentacle moves, you can record from the neurons inside the tentacle, or you can record around, touch the neck, where all the tentacles come out, and then present the tentacle with food, and like you can sometimes record a potential. But that being said, I've tried a lot of times, and I've never gotten into it.
Is that the Nobel Prize you're talking about?
I think there's probably once the group that figures out neural nets is probably it'll be like the squid or the amnesia, the Aplysia. It'll be like some that kind of thing going on.
How big are the anemones that you've worked with previously?
Big, yeah. And then like this big.
So Danielle, what's your main research focus right now? I know that you're presenting some of the stuff that you've worked on, but what's your current goals?
Okay. What I am working on now is in evolution, but of morphology. I am working on how muscles in the foot of birds during embryology promotes the acquisition of the configuration of the skeletal elements in the foot during embryology, but to infer how was the evolution of this configuration from dinosaurs. So that is my so topic cool. Now, but Tim knows that I like so much general different topics about biology. One of the topics that I like so much is this early evolutionary view of the animals, and other things are endosymbiosis. Endosymbiosis I like so much endosymbiosis because it's another way to understand how evolution was. And other things that I like so much is dissemination of science. In general, I have been working on working in different activities to teach how microscopy works. So I like a lot of fields. So I told you that if you want to know more about these fields, or maybe about multicellularity, the origin of multicellularity, or some characteristics around about eukaryotic cells, single cells organisms like Protista. You can write me, because there are a lot of things that I didn't show you in this talk, because obviously I need to finish sometime. But there are a lot of interesting things in Protista. Actually, I didn't show you, but Protista have eyes sometimes. They develop a kind of eyes in a group named Barnovia. They develop a kind of eyes. They have rhodopsins. They have homologous of other proteins that we find in animals. It's very, very interesting. So, yes.
That's incredible. that's really cool. Thank you so much for the talk. I'm going to get back to work, but thank you for everything. And I will be in touch.
Okay, thank you, and thank everyone. I think that I need to go out now because I need other things to do also. But if you want to keep in touch, you can write me to my email, okay? Okay,
thank you so much for your time, Daniela.
Thank you.
Bye-bye.
I enjoy a lot. Bye-bye. Thank you.
Ciao.